Hello everyone, I am Dr. Sonali and today we will be talking about vaccines. So this whole lecture of vaccine I have divided into two parts. In the first part, that is this part, we will be talking about the mechanism and the types of vaccine. In the second part, we will be talking about the cold chain and the vaccine vial monitor, the newer method of vaccine, that is the new technologies of vaccine and the national immunization schedule. Okay, now let's see the learning objective for today's lecture. First, we have the introduction. Then we have the difference between active and passive immunization. Then types of vaccines with example. Okay. Now, let's see what are the important questions from this topic we have. And now, before that, why is this topic important? So, be it any course, be it your MBBS or your nursing, your BDS, your MD, that is MD Microbiology, this vaccine topic is very important because you get lots of short notes. So, in short notes, you can get your live versus kill vaccine difference. You can get your subunit vaccine. You can get passive immunization. You can get cold chain and vaccine vial monitor. And you can get newer vaccine technology, that is the mRNA vaccines and edible vaccine, and also the national immunization schedule. Now, as a long question, you can also get the definition, what is vaccine, combined with types of vaccine and national immunization schedule. Okay. For your NEET PG exam, lots of MCQs also come from this topic. Okay. So let's start. Now, before starting, let's see this definition first. What is immunoprophylaxis? It is prevention of infectious disease via immune system. If your immune system is an army, it is an army, right? Your immunoprophylaxis can be considered as a training drill. Okay. Now, how do you train your army? You train your army in two ways. Either actively by stimulating own immunity, that is by vaccine. Second, passively by giving ready-made antibodies in the form of immunoglobulins. Okay. Now, let's talk about active immunoprophylaxis. Before we go into detail, let's talk a bit about the history. Now, you know the term vaccine is derived from variole vaccini. This term was given to us by a legendary scientist, Edward Jenner. He used this term for cowpox. He came up with this legendary idea to use this for the treatment of smallpox, to cure smallpox. Okay. Now, this idea was given by Edward Jenner, but the name vaccine was given to us by Louis Pasteur. Okay, this was about the history. Now, let's see the definition of vaccines. What are vaccines? Vaccine is an immunobiological preparation that provides specific protection against a particular disease. So, it's like a mock drill. Okay? It's like a mock drill, that is, it trains your immune system to fight against an organism without the actual chaos of that infection. Okay? So, what all it does, it triggers your B cell, that is by producing antibodies, and also T cell, that is cell mediated immunity. Now, it also triggers your memory cell. Because of this, there is long lasting protection. The example of active immunoprophylaxis include hepatitis B and your measles, mumps and rubella vaccine. Okay. Now, let's talk about certain principle for your vaccines, that is immunoprophylaxis. Now, your vaccines have certain targets or valencies. Based on this, your vaccine can either be monovalent. Now, it means that your vaccine has only one serotype. Okay. A very good example of this is your hepatitis B vaccine. Now, your vaccine can also be polyvalent. Polyvalent means it has more than one serotype, multiple serotype. A very good and common example of this is your seasonal influenza vaccine. Okay. Now this was the target or valencies. Let's talk about your vaccine being homologous or heterologous. Okay. Now, homologous means the vaccine contains the same organism. The vaccine is derived from the same organism which it is targeting. Clear? Heterologous means the vaccine is derived from a different organism than the one it is targeting. It is derived more from a cousin organism. A very good example is like the one we saw, cowpox used to treat smallpox. Now this idea was first used by Edward Jenner. So this is also known as Jennerian vaccine. Okay. Now another very good example of this is your BCG, which uses Mycobacterium bovis for MTB. Clear? Now let's talk about passive immunoprophylaxis. Now imagine your immune system is down and at the same time your villains or your microbes just walked in. Now you don't have time to train your army. Okay, because your immune system is down, you cannot train right now. So what you will do in such cases, you borrow soldiers. This borrowing of soldier is known as passive immunoprophylaxis. Okay. So what do we do in passive immunoprophylaxis? We give preformed antibodies known as immunoglobulins. Example, your anti-rabies immunoglobulin and anti-tetanus immunoglobulin. Okay. This preformed antibodies, that is this ready-made antibodies, are given in a vial. So it has some advantages also, some disadvantages also. The good part about it, there is no B cell or T cell stimulation. Okay. It is a fast immunity. It works very fast, but it also doesn't stimulate memory cell. Because of this, it's not long lasting. So it is immediate but short lived. Now in what all situation, that is when do you use this passive immunoprophylaxis? It is used in exposure, which we call as post-exposure prophylaxis. That is when you encounter certain organism after which you directly need ready-made antibodies. You don't have time to wait and train your immunity. Okay. So the examples are rabies, hepatitis A and B, tetanus, measles, your varicella zoster virus infection, cytomegalovirus infection, and rubella. Okay. Then you can have to use your passive immunoprophylaxis in an immunocompromised case. You know, when the patient is on steroids, and the patient has chemotherapy, or when the patient is suffering from a disease like your AIDS. You can also use passive immunoprophylaxis for toxin-mediated disease. For example, your Corynebacterium diphtheriae. Okay. So when this bacteria enters our body, it releases toxin. Okay. Now, when you give antibiotic, what happens is it only targets the bacteria, but it doesn't target the toxin. So what do we require? We require preformed antibody for these toxin. Okay. So we give antitoxin here, targeting the toxin. Clear? Now, let's see the difference between your active and passive immunity. So, your active immunity, its immunity develops slowly, days to week. Passive, it works instantly. Post-immune system for active immunization, yes, it must be functional. For passive, no, even if defective, it's okay. Memory formed for active immunization, yes, memory is formed, but for passive, no, it's not formed. Duration of protection for active immunization, it's long term, and for passive, it's short term, weeks to months. Now, let's see in the active immunoprophylaxis, the types. That is, first is the live attenuated vaccine. So, let's see what this vaccine contains. It contains the organism which is live, that is alive. Attenuated. Attenuated means it has been made harmless or weakened. Now, how do you make something weak or harmless? You make something weak or harmless by putting that thing or organism here in an uncomfortable or unfavorable environment. Same goes with a human being. How do you break a human being or make that human being weak? By putting that human being in an uncomfortable or unfavorable situation. That is your attenuation. It refers to reduction of virulence which can be achieved by passing through unfavorable host, repeated culture in artificial media, growth in high temperature or in presence of weak antiseptic or prolonged storage in cultures. Now, let's see what are the examples of a live attenuated vaccine. Well, it's a long list. So, I have come up with a mnemonic. Whenever you remember your live attenuated vaccine, always remember this. Have you ever seen the preparation of live biryani? Okay. How is the live biryani made? You need oil for that. You need veggies because it's a veg biryani. Then you need salt. You need masala. You need rice, obviously. Then you need turmeric. Put it all in a hot button. So let's see the example. This live here stands for live attenuated influenza vaccine. Clear? This stands for BCG. O stands for oral typhoid vaccine. Okay? V stands for varicella zoster virus vaccine. S here stands for Sabin vaccine, which is for oral polio virus. This M stands for measles, mumps, rubella. R here stands for rotavirus. Now, what is the color of your turmeric? It's yellow. So, it's for yellow fever vaccine. The H here stands for hepatitis A and B here stands for Japanese B encephalitis. This is your list of live attenuated vaccines. Now, let's see some of the advantages that your live attenuated vaccine have. The first advantage it has, it multiplies in the host. How is this an advantage? Since it multiplies in the host, there will be more antigen. As there is more antigen is available, there will be a strong immune reaction. Okay. Now, another advantage is that live attenuated vaccine retain the immunogenicity of the organism. It retain even the major and the every minor detail of the organism. So, what will happen because of this? There will be a stronger immune reaction. your live attenuated vaccine also stimulate mucosal immunity. Now, your mucosal immunity is also known as local immunity. It is IgA mediated. Okay. Now, we'll see through an example that how can it stimulate local immunity. For example, your oral polio virus. Now, you know your oral polio virus, when it goes in the intestinal mucosa, what it does, it stimulates the production of IgA. So the IgA covers the intestinal mucosa and prevents the entry of your polio virus locally. So it's a local immunity. Clear? Now let's see the disadvantages of a live attenuated vaccine. It cannot be used in immunocompromised patient, okay, and in pregnancy. Why in pregnancy? Because there is a risk of vertical transmission, right? You can use in certain cases when the benefit is more than the risk. Clear? Now let's see some points to remember for your live attenuated vaccine. If 2 live vaccines are needed, a minimum of 4 weeks gap is required, except your yellow fever and MMR combo. Usually 1 dose gives you long-lasting immunity, except your oral polio vaccine, it needs multiple doses. Remember that ad that used to come, do boon zindagi ke, it was more than 1, so multiple doses. Risk of reverting to virulence if the attenuation, that is making it weak and harmless, isn't perfect. So what will happen? It can revert back to its virulent state. It needs strict cold chain, especially your OPV and measles. And among these two, your OPV is extremely the most heat sensitive vaccine. Okay, so this was your live attenuated vaccine. Now let's understand it through a diagram. So also you can have a revision through this. Your live attenuated vaccine is delivered by syringe or by mouth. Vaccine virus enters the cell and replaces coat to replace itself. Now instructions are read and cell makes more virus. Okay, and this replicated weakened virus is replicating here. Now this replicated organisms are taken in by the antigen presenting cell. Now you see the immune helper cell triggers on the immune response after seeing this on the antigen presenting cell. Your body creates army of B cell to produce antibody against this virus or any organism, be it bacteria also. So antibody bind to the virus or organism and prevents entry into the cell. Your memory cells are created to launch an attack if the body encounters this organism, and infected cells are then recognized and destroyed. So the points to remember here is the weakened organism replicates and memory cells are formed. I hope that was clear. Now let's talk about inactivated vaccine. Now your inactivated or killed vaccine contain organisms which are inactivated or killed. It's like your actual organism, the actual villain is dead and you are using a duplicate. Seeing this duplicate, your immune system will still produce antibody. Your immune system will still be trained. So how do you make this killed vaccine or inactivated vaccine? The organism is grown in culture, then killed using heat or formaldehyde. Okay. Now for inactivated or killed vaccine, let's see some points. Is it safe? Yes, it's very safe. It's actually safer than your live vaccine. Why? Because there is no risk of reverting back to virulence. It's already killed. Second point, does it work fast? No. Not fast as your live vaccine. Now, is the memory cell formed? No. Memory not formed. Okay. Also, it doesn't multiply. No multiplication in the once it is delivered in your body. No multiplication. Okay. So, because of all of this, for your inactivated or killed vaccine, you need booster. and also adjuvant to be given with it. Why? Booster to maintain the immune reaction. Okay, to maintain a strong immune reaction, you have to give booster. And adjuvant, why? It enhances your antigen uptake, and adjuvant also increases your antigen exposure time. What happens because of this? Same strong immune reaction. Okay, because see, apart from being safe, it has certain disadvantages. So to counter these, what do? We add boosters and adjuvant. Clear? Now let's see certain points. You see, we can give the inactivated or killed vaccine by intramuscular or intradermal. We don't give it orally. Okay? And the only contraindication to inactivated or killed vaccine is if the patient had a previous reaction to it. Clear? Now let's see the example of inactivated or killed vaccine. Now again, it's a long list, so I've made a mnemonic for it. Whenever you have to, memorize the inactivated or killed vaccine, remember Japan. In Japan, everything is clean. Their home, their kitchen, everything is very clean. So I asked from a person of Japan, how do you keep your kitchen clean? So he said, I know to clean pots, pans, and heavy racks. Okay, so let's see the example. This is your Japanese bee encephalitis. Clear? Now see, that Japanese bee encephalitis was also in your live vaccine. That was a different strain. This is a different strain. Okay, I is injectable polio vaccine. K is killed influenza vaccine. T stands for typhoid vaccine. C is for cholera vaccine. P here stands for pertussis. P in pan stands for your plague vaccine. The H here stands for hepatitis A, and R stands for rubella. Sorry, R stands for rabies. Not rubella, R stands for your rabies vaccine. Okay, so this was all your example for your inactivated or killed vaccine. Okay. Now let's move ahead. Let's see a diagrammatic representation. So your inactivated vaccine is delivered by a syringe. The inactivated vaccine organism is taken, inactivated organism is taken up by the antigen presenting cell. Now the point to notice and remember here is the vaccine is chopped into pieces called antigen, and the inactivated organism cannot replicate. So the antigen is displayed on the cell surface where they can be recognized by the immune system. Okay. Now again the immune helper cell triggers the immune response. Body creates army of B cell to produce antibody against the wild organism of the wild virus or bacteria. Now your antibody binds to virus or bacteria and prevents entry into the cell. Now there is no memory cell boost. So the antibody levels fade with time, and usually additional shots are needed, that is boosters are required. Okay. So let's quickly fill this together so that you can revise also your killed and live vaccine difference. Number of doses. more in killed vaccine. In live vaccine, it's mostly single, except OPV. Clear? Need for adjuvant is in killed, not in live. Duration of immunity, more in live, it's less, it's short in killed. Effectiveness of protection is more in live, it's less in killed. Mucosal immunity is stimulated in live vaccine, not in killed vaccine. Reversal back to virulence is true for live vaccine, not for killed vaccine. Fecal-oral spread. Now see, anything which is live, that can only spread through your fecal-oral route. So it is true for live vaccine, not for killed vaccine. Interference by other microorganism in host by live vaccine. Only you can interfere with something which is alive, not for killed vaccine. Stability at room temperature is more for killed vaccine, is less, or not at all for your live vaccine. Immunodeficiency in pregnancy, you can give killed vaccine, you cannot give live vaccine. Okay, it is contraindicated. Clear? Now let's talk about your subunit vaccine. See, your subunit vaccine is not made from the whole organism. It is made from only one part of the organism. Okay, so let's see the definition. Subunit vaccine are made from a piece of a pathogen, not the whole organism. So they do not contain any live pathogen. Some important subunit vaccines are your polysaccharide vaccine, conjugate vaccine, and your protein-based vaccine. See, the types are based on whichever part of the organism they are made. So if it's made from a polysaccharide part, they call it polysaccharide vaccine. If from a protein based, then a protein-based vaccine. If from a combined part of both, it's a conjugate vaccine. So let's see all of them one by one. First is your polysaccharide vaccine. They help you make protective response against the surface of organism, allowing your body to kill better. For the response against the surface of organism which contains sugar. Okay, that is polysaccharide. It is mostly in the form of a capsule. Okay, a very good example of this is your PPSV 23, pneumococcal polysaccharide vaccine. Now, this vaccine is very good vaccine for adults. But when it comes to children less than 2 years of age, it's not a good vaccine. It's less immunogenic. Okay. Now, we have a problem here because your pneumococcus affects less than 2 years also. So, now we require to add something in it. That's when we have the concept of conjugate vaccine. In conjugate vaccine, you have a sugar, you add it with a protein component. Now, the protein component here acts as an adjuvant. And we have already seen a function of adjuvant. It enhances your antigen uptake and also increases the antigen exposure time. Okay. So, because of this, it becomes more immunogenic. Okay. So, since it's more immunogenic, so what will happen now? We can use it for kids less than 2 years of age. Now, another example of this is for conjugate vaccine. One example is PCV 13. That is your pneumococcal conjugate vaccine. Another example, Haemophilus influenza type B vaccine. Okay. Now, we have a protein-based vaccine which is made from the protein part of the organism. Now, how do you make these subunit vaccine? You can either make it from the whole organism, or there is a technology known as recombinant technology. We'll see how it is made with an example, how this recombinant technology works with an example. One example of your subunit vaccine is also your hepatitis B vaccine, which uses hepatitis B surface antigen. Now, with recombinant technology, what we do is we take the gene coding for your hepatitis B surface antigen and put it in your baker's yeast. Okay. So, as your yeast is multiplying, so this gene is also replicated. And once enough of, we have a mass production of this gene, we can use it for vaccine. Okay, so this is known as recombinant technology. Now let's have a diagrammatic representation of it to understand it better. You see, this bacteria, this has polysaccharide. So in this vaccine, you can either use the polysaccharide or you can use the conjugate part. That is, add polysaccharide with protein. So it's conjugated, joined together. So this vaccine is released into the body. Vaccine is taken up by the antigen presenting cell and displaced on surface at the antigen. Threat detected, and immune helper cell identify antigen and trigger an immune response. Okay. Now let's see toxoid vaccine. Now, what is toxoid? Toxoid is a form of toxin which had lost its virulence. It has lost its virulence, but somehow it has still maintained its immunogenicity. In simple word, you can say that it has lost its power to attack. Okay, so it has lost its power to cause any harm, but it can still stimulate your immune system. Clear? So it can still train your immune system. Toxoid vaccine use inactivated toxins to target toxic activity created by the bacteria rather than targeting the bacteria itself. Okay. So here we can, see it like this. Toxoid vaccine is created from toxin. So how do we create it? By detoxification. How do you detoxify? In acidic pH, formalin, or by prolonged storage. Okay. Let's see what I was talking about. So this wild bacteria, this produces a toxin. So by detoxification, we have made this harmless toxoid. Okay. Now, what happens is this toxoid vaccine is released into the body. There is this harmless toxoid. Now you see, this toxoid cannot bind to the cell as this bacterial toxin do. This bacterial toxin, it enters and eventually destroys the cell. Now this property, this virulent property is not present in the toxoid. So this is not possible. But toxoid can trigger the similar immune response as bacterial toxin by going to antigen presenting cells, and the immune helper cell triggers an immune response. Body creates army of B cell to produce antibody against them, and antibody binds to the toxoid or toxin and mark for destruction. Antibody levels fade with time, so usually additional shots are needed. Clear? Now, let's understand something. We just talked about antitoxin also. So let's quickly see the difference so you don't get confused. Antitoxin is a preformed toxin. Okay, it's a preformed antibody for your toxin. Whereas toxoid is a chemically modified. It's a chemically modified toxin which trains your immune system to produce antibody against toxin. So this one is example of passive immunity. This one is example of your active immunity. Okay. Now let's understand this with an example. Say, for example, your Corynebacterium. Okay, this causes diphtheria. So for your Corynebacterium diphtheria, if you are just trying to prevent this, for say in a newborn, you just want to prevent this right now. So what you will do, you will give toxoid. Okay. So you give vaccine in the form of toxoid. So this is for prevention. But imagine somebody is already infected with this Corynebacterium. So what do? We give antitoxin. Okay, we give antitoxin. So this is for treatment. I hope this was clear. So this first part of lecture ends here. In the second part, as I said before, we will be covering the cold chain, vaccine vial monitor, newer vaccine technologies, and the national immunization schedule. Thank you.